All right. The next major unit that we did after scientific method was the biochemistry unit. You really need to know about 4 kinds of compounds. There's carbs, proteins, lipids, and nucleic acids. Now, lipids, you really don't need to know for the regions. So we're going to just ignore those for this review. But the others come in a small unit that repeats and a large unit. Okay, and I've put here in the table the large units. Carbohydrates include starch, cellulose, and glycogen. Starch is the most tested. Okay, polypeptide, or sometimes called a protein, simply when it's in its large form. Okay, so you are going to take a moment to try to remember what the small unit is. So our carbohydrates in the large unit, if you eat starch, when you digest it, you're going to break it down into whatever goes here. When you eat a protein or a polypeptide, when you digest it, you break it down into its small unit right here. And in fact, we also talked about this unit here because we talked about when proteins are made in the ribosomes, these get strung together to make the protein. And then finally, we have nucleic acids, and that's DNA and RNA, and what we call the little subunits that make up those strands. So let's see how you did. Carbohydrates, starch, if you digest it, it gets broken down into sugars, particularly glucose. Polypeptides, or proteins, are broken down into amino acids. Remember, we talked about the tRNAs carrying the amino acids over to the ribosome to string them together to make a protein, right? Amino acids make up proteins, and if you eat proteins, you digest them down into amino acids. And then finally, nucleic acids have nucleotides. That's the little circle, pentagon, and rectangle together. So those are basic compounds that you need to know. Really, really important to know that carbohydrates include starch and sugar, and that if you digest starch, you get sugar. If you digest proteins, you get amino acids. And DNA and RNA are made of nucleotides. And just as a little connection, recall that the system that is involved in breaking down starch into sugar or protein into amino acids is your digestive system. And that's necessary because big things won't fit across your cell membrane. Little things will. All right, some more from the biochemistry chapter that's really important to know. pH. pH is really important because it affects the enzymes in your body. pH is a measure of how acidic or basic something is. The numbers below 7 are acids. The numbers above 7 are bases, and 7 is neutral. The quick and easy way to remember that is write 1 through 14. Write down next to you as you're taking the test. 1 to 7 comes first. 7 to 14 comes second. A comes before B. Acids are the low numbers. Bases are the high numbers. Gets tricky because if they ask you about a strong acid, that's a low number. If they ask you about a strong base, that's a high number. So something that will eat through corrode metal because it's so acidic is going to be like a 1. All right. Critical. I put it in bold because you have to know about enzymes. You wouldn't be alive without enzymes. Every chemical reaction in your body, whether that's respiration, breaking down sugar to get energy, or if it's building proteins at your ribosomes, or any chemical reaction that we talked about, it's controlled by enzymes. If you waited for them to happen without enzymes, you would die long before you had enough energy. So these are really critical, okay? They are proteins that speed up reactions without being changed. Anything that controls a reaction and speeds it up without being changed is a catalyst. Now, our particular ones are proteins, so we'll call them enzymes. On the exam, if you see the word catalyst, it's interchangeable with the word enzyme. They just go back and forth. Okay, but an enzyme is specifically a protein that is a catalyst, and catalyst is a broader term. But for biology, either of those terms are interchangeable with the other. Okay, the way that enzymes work is they fit exactly to their substrate, like a puzzle piece. Oops, they fit like a puzzle piece. So an enzyme that digests starch into sugar is going to have a shape that fits starch. It will not fit proteins. It will not digest proteins. It will only fit starch. It will digest starch. Okay. So the shape of it is critical. And there are some things that affect its shape. One is pH. If you have an enzyme that works well in an acid and you drop it in a base, it'll change shape and it'll no longer do its job. This is why it's so important that you keep the right pH in your cells. Another thing that will change its shape is temperature. If you make an enzyme really, really hot, it will change shape. It's called denaturing. It just essentially falls apart and it will no longer fit its substrate and it will no longer do its job. This is why if you have an extremely high temperature, your enzymes may stop working and you die. Now, those are really hot temperatures. You have to be really, really high for that to happen. But it's really important to all living systems that the temperatures be in the right range, the optimum temperature for the enzyme or the optimum pH. Most of the enzymes in humans, their optimum temperature is 98.6 degrees Fahrenheit, 37 degrees Celsius. So, our entire body is that temperature because those are the best best temperatures for our enzymes. We have enzymes that work at different pHs. The ones in our stomach need an acid. The ones in our intestines need a base. And then how much enzyme you have will affect enzyme activity. So this is how it comes into being tested. Since enzyme activity is affected by pH and temperature, so is everything that you need to be alive, including respiration and photosynthesis. So they may ask a question about respiration or photosynthesis and say they're changing the temperature. And why does that affect it? It's because it changes the shape of the enzyme.